This is episode 227 of the Stem Cell Podcast, Interspecies Chimeras, with Dr. Jun Wu. Hey, everybody, we are Daylon and Arun. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today, we have Dr. Jun Wu from the University of Texas Southwestern Medical Center. He's on the podcast to talk about his work using interspecies chimeras to study fundamental biology. We've also got our usual roundup of recent highlights in stem cell news coming up.
But first, we'd like to remind our listeners about Organoid News, a free weekly newsletter summarizing the latest research news, jobs, and events in organoid research. Use Organoid News to stay current with the latest applications and discoveries using organoids. Subscribe for free at www.organoidnews.com.
All right, Arun, I'm kicking off with something came out of a locker room. I mean, this is a little bit out of left field, talking about Aspergillus fumigatus. We don't usually talk about fungus on the show. It's a cell show, but fungus that has cells too. Newsflash. And Aspergillus is ubiquitous in the air and soil, and we inhale several hundred spores per day. And that's several orders of magnitude higher in environments like construction sites or agricultural sites. And typically, in a healthy individual, the spores are mechanically removed by airway cilia and expectoration, and also immunologically by the alveolar macrophages and other innate and adaptive immune cells. But in immunocompromised patients, you can have these spores persist, and it can result in local pulmonary pathology. It's called invasive pulmonary aspergillosis. I mean, this is troubling. It's germination of the spores in the lungs, some alien type parasitism there, and then inflammatory response downstream of that. And it's typically observed in severely immunocompromised patients or recipients of intense chemoradiotherapy or following allogeneic hematopoietic stem cell transplant. But the prevalence of IPA is increasing because there's these new therapies, these tyrosine kinase inhibitors, where it's like a off-target effect or a side effect. And more than that, you have a high incidence of IPA in patients with pre-existing or concomitant influenza, cytomegalovirus, and recently SARS, right? Coronavirus is becoming really relevant, even though maybe Corona is on the downswing, but it's not going anywhere. It's kind of endemic, I would speculate now. So in the context of COVID, you can get prolonged hospitalization, extended ventilation support, and excessive morbidity and mortality. So yeah, it's an issue. This IPA and Aspergillus being the root needs to be addressed. But conventional antifungal therapy, whether or not it works in the context of, I mean, we know it works for Aspergillus with like athlete's foot, right? But in the context of IPA, unknown. All right, so here's where we come out of left field. But, left field is very well populated these days in the CAR T world. This is a story from Michael Hudacek and Jürgen Löffler. Who they're from Würzburg, Germany, doing my best there with the pronunciations. They had a story in Science Translational Medicine where they tried to leverage CAR T to attack Aspergillus, right? They gene engineered Aspergillus fumigatus specific CAR T cell and showed that it recognized a conserved protein antigen in the cell wall of the hyphae. So, I mean, this is like surgical. And then in an in vivo model of IPA in immunodeficient mice, they show that these CAR T cells localize to the site of infection. They reduce the fungal burden in the lung. And when you did adoptive transfer of those cells, it was even better with the antifungal efficacy and resulted in overall improvement in survival. So, I mean, look, I don't know if anybody's going to be getting in some CAR T for IPA on event, right? Or anything like that. But, I think this is, I think, and maybe they will. There's some persistent infectious fungal diseases, I'm sure that would be well suited to this, but let's say that everybody's not going with their classic antifungal approach using CAR T. I think the real Trojan horse here is how they're using CAR T for everything. You know, when you're going after it for this fungal disease, I think all infectious diseases are on the table. We've talked about fibrosis previously on the show. We're going to circle back around to that in my second roundup story. But it looks like, you know, there's just an endless list of targets that are going to be addressed by CAR T. Really, the therapy of the century, if all of these targets pan out, being, you know, practical.
Yeah, the potential is unlimited for something like this. I mean, it's not just infectious disease, it's not just disease, period. It's anything that has a presenting antigen that could be targeted by a modified CAR T. If you need to clean out some tissues in some situations of fibrosis, you can do that. You can have a really unique application here with the antifungals. It's tremendous, like what you're alluding to. I mean, the fungus is absolutely among us, as the kids say. But I think for the most situations, what we were saying here, you know, antifungals will do their job, but perhaps in extreme chronic cases of, you know, persistent fungal infections, this could be a really unique approach. Also, the fact that you mentioned that tyrosine kinase inhibitors have this potential off-target effect of causing IPA, that's terrifying to me as somebody who studies tyrosine kinase inhibitors in another context, which they cause cardiotoxicity in a lot of situations. So, you know, these drugs have a lot of off-target effects, and this is not a pretty one. This is I definitely do not want to experience this in my lifetime. That's for sure.
Yeah, I mean, as someone who suffered from asthma downstream of mold as a youth, I mean, it's debilitating, and that was mild. So just the idea of anything colonizing my lungs and germinating, yuck. You may argue that we've jumped the shark maybe a little bit here. We're using CAR T for antifungal, but as you said, there's a real broad range of really serious fungal conditions that could be addressed by this. And as we've been saying, this is really just the tip of the iceberg in applying CAR T to everything under the sun.
Absolutely, and I think you've got a little bit more in this area in your next roundup paper, but we'll stick to a blood adjacent story. We're talking about CAR T, and we're going to move on to more of a, I guess, a developmental biology story about the blood. This is coming from the lab of Len Zahn in Boston at Harvard. Uh, this is, of course, what Lenzon does and has been doing for a long time, which is studying hematopoiesis using zebrafish as the workhorse model system in their laboratory, combined with amazing imaging technologies that you can utilize in conjunction with zebrafish, of course, because they're very amenable to imaging, being transparent and small and all these things, and genetically manipulable, very easy. So the title of this paper, which is actually it's a science paper, is "Quality Assurance of Hematopoietic Stem Cells by Macrophages Determines Stem Cell Clonality." First author here is Sam Watrous, and again coming from Len Zon's lab. So we've been obviously, talking about HSCs for a while on the show, and it seems like every single episode we have some sort of HSC story here. But this is unique because it reminded me of a story that we covered a while back about the macrophage cuddle. I don't know
Mm.
if you remember that particular phrase that kind of stuck with me. That's exactly what's going on in this situation, but in sort of a different context. Apparently, these macrophages are cuddling these, hematopoietic stem cells during development. So we can dive a little bit deeper into it. And there's a specific reason why cute interaction is actually happening. So we know that these tissue-specific stem cells, HSCs, can persist for a long time, can differentiate to maintain homeostasis, or even in severe situations turn into cancer cells. This is the unfortunate reality of blood stem cell biology. But what about quality assurance? How do we make sure that these cells, these HSCs, hematopoietic stem cells in particular, are okay? They're high quality and they're maintaining their high quality over the course of a lifetime. And in particular, new HSCs, newly formed blood stem cells, how do we ensure their quality as well? So, what they did here is again using some amazing imaging, live imaging, multiple genetic crosses here in zebrafish. They observed, in their words, not mine, quote, "intimate and specific interactions between macrophages and early blood stem cells in zebrafish embryos." So again, very cute. And there's very cute images in this paper as well. So these macrophage interactions led to either the removal of the cytoplasmic material and stem cell division of the HSCs, or actually complete engulfment and death of the HSCs. So they're doing a regulatory function. These macrophages are in the context of these HSCs. So they used a cellular barcoding strategy and actually found out that a particular gene in protein calreticulin, when knocked down or depleted, actually reduce the number of stem cell clones that establish adult hematopoiesis. So they actually, that's a really cool thing. They found a specific protein, a specific mechanism that when you regulate it can actually mess with this cuddling mechanism a bit. So I think this is mind blowing. Another really cool application for macrophages early during development. Certainly, we have to consider the species specific side of this and the evolutionary conservation of this particular mechanism, but really cool. And I think something that can only be enabled by some of the amazing high resolution imaging that you can do in zebrafish and that they're also doing in the Zon lab for a long time now. So really cute, very unique study in my opinion.
Yeah, you got to respect the macrophage. A lot of unappreciated biology going on there. Um, but yeah, I'm glad you introed with that whole cuddling thing because that's what jumped out at me. I remember it was like a decade ago, legitimately a decade ago. I got a paper to review when I was, still working closely with my mentor. They don't send lens on papers directly to me. I'll tell you that much. But I got a paper to review. It was about the cuddling thing. And I was like, "Oh man, Len, with this anthropomorphizing of the biology and all that." And I thought it was like, and my critique was, well, it seems a little bit descriptive. And I was stuck on this idea of like, why are you calling it cuddling? Like, honestly, I'd be happier at that point. I would have been happier with the intimate interaction. That seems a little bit more scientific. But this, I think, is to Dr. Zahn's credit, is why he's such a admirable and inspirational scientist. I think is because that's he starts with an observation, and I think that makes it really accessible to scientists of all levels. Is it really it's not in the weeds of the language and the biology. It's something really that jumps out at you, just an observation there, cuddling, and now here we are a decade later. He's taken it all the way. It started with that, and then as you spoke about the imaging took it to another level, but now with the cellular barcoding, he's really made an observation and then connected it to a real why, how biology question that has tremendous impact and importance and relevance to disease and health and mechanism. And so, you gotta love Len for his approach and yeah, the way he makes these ideas accessible to people by talking about things like intimate contact. But I mean, at the end of the day, this is an important story that was unearthed after a bit of a cuddle.
Much respect to one of the gurus of HSC biology and Len Zahn, and of course the amazing work that his lab did in this particular paper. But you make a really good point. I mean, part of the reason that certain papers make a splash, and part of the reason that they take off, is because of good science communication, which hopefully is something that we, espouse and something that we encourage here on the show. Is good science communication, better than others sometimes. But yes, this is a very important skill to learn no matter where you're working, what you're working on is how you can adequately convey the studies and the discoveries that you make. Another example of something like this is the CD47 protein, which
Mhm.
was really worked on very heavily by the folks at Stanford and Irv Weissman's lab. The quote is always the "don't eat me" signal, right? So again, a very simple analogy, a very simple phrase that sticks with you. I still remember that. I don't do anything close to CD47, but I still remember what that particular protein, what that signal does. It's the "don't eat me" signal, just because of that really important phrase, and that's a testament to good science communication. Again, right there. So I guess yes, absolutely fantastic paper and a good reflection on the importance of good science communication.
Absolutely. And we learned that during the pandemic, right? This isn't just among scientists who have their own language, but really at the end of the day, it's about getting the message across to the public so we can move the needle. In the case of the pandemic, it was about vaccination. That's a segue to my story here. And I mean, this might get the anti-vax crew a little riled up, but don't go crazy, people. No one's proposing to do this tomorrow. But I think the impact of this, like CAR T, is I mean, commensurate with CAR T potentially, but I think a lot more accessible as an idea. You know, as you were alluding to the "don't eat me" signal, the idea of vaccination is basic. You train the body, and then it goes out and seeks out antigens and shuts it down. Usually in the context of disease, right? Of invaders, infectious disease. But here, this is a story like the CAR T attacking fibrosis, a story about using vaccination to address fibrosis in the liver and the lung. So, you know, fibroblasts are critical, important for maintaining the structure of any solid tissue and homeostasis in that tissue. But there's this expansion of activated so-called fibrogenic fibroblasts and this maladaptive response in response to uh, chronic tissue damage or injury that results in organ failure by depositing all this extracellular matrix and this kind of, again, maladaptive, non-productive fibrosis. Um, and the global health burden of fibrosis is huge, specifically in the liver and the lung. These are major issues. And there aren't many therapies. I mean, underscored, the unmet burden is huge. Now, in terms of how we can address this, we alluded to this earlier in the show. There have been studies that use genetic ablation of fibrogenic cells to reduce fibrosis in mice, and this is using genetic engineering of mice. Not really feasible at all in a clinical context, but at least it says two things. One, that fibrosis can be addressed, and if it is, it can improve the health of mice in this case. But also, the fact that you use these genetic approaches in mice show that there are genes that specifically tag or mark fibrogenic cells, right? So based on that, there's been multiple approaches using CAR T, as we just talked about a bit, but engineering CAR Ts to eradicate fibroblasts. That was based on fibroblast activation protein, or FAP, in the heart. We talked about that on this show. Also, senescent cells that express urokinase plasminogen activator surface receptor, or UPAR, that's been used in the context of liver fibrosis. But those are cell-based therapies, and I think those have a lot of legs, frankly. I mean, I might go with CAR T before I went with this approach, but it's always nice to have another arrow in the quiver. And this group, which was led by Christian Stockman in Zurich, Switzerland, reasoned that the proteins encoded by genes specifically expressed in fibrogenic cells can result in these self-peptides on the surface of fibrogenic cells that can be targeted using a more vaccination-based approach. T cells that are in the body, not that are integrated into the body or introduced into the body after engineering. So basic immunization. And that's what they did. I think it was a pretty impressive approach. I would have thought that'd be a lot riskier, but it seems to have worked. They used in silico epitope prediction to identify genes that would drive self-peptides, and then they use those self-peptides to immunize these mice and demonstrated efficacy of this vaccination approach to mount a CD8 positive T cell response that reduced fibroblasts and fibrosis in both the liver and lungs of mice. So, I mean, seems so straightforward. You talk about getting a modRNA injection for vaccination against COVID. That was pretty easy. Could we do the same for fibrosis? I'm not sure. Arun, what's your take on this?
Yeah, I think it's again similar to the previous study that you covered. The application, if this pans out, is so unlimited because, as you're alluding to, fibrosis is something that's almost universal across different disease states. Of course, you have fibrosis of the heart after myocardial infarction, and liver fibrosis as well. So I think there has to be a lot of work to validate this, especially in a human setting. But really, it's really a very cool approach, and it makes me think of some of the other novel vaccination-based approaches that we've been talking about recently. I'm sure you remember that tumor vaccine that folks have been working on. Even my former mentor, Jun Wu, had this iPS-based tumor vaccine of where you can introduce certain iPSCs where they're expressing a certain antigen and can serve as a quote vaccination strategy against cancer. So I think calling something a vaccine, it's not a vaccine in the traditional sense, perhaps, but I think the approach and the downstream is similar. So I think the applications are really unlimited here. I think there are a couple of things that they mentioned that they still need to do, and I'm kind of surprised why they didn't do this. For one, they didn't do any single cell analysis to gain some more insight into the mechanisms of the fibrosis regression and liver regeneration. To me, that is really important in part because we always talk about single cell on the show and how accessible it is, but also because there are so many different populations in the liver that are critical to the regenerating liver and the cross, cellular interactions there. So I think that would be in my mind, that would be a pretty easy experiment to do, and I'd be shocked if they weren't already doing that. So they probably covered their bases right there. But yeah, I mean, I think a really cool, perhaps universal approach to addressing fibrosis.
Yeah. Critically important though, as you said, to look at the what's going on in that liver because, I think what we're all, you know, ignoring the elephant in the room here. We're vaccinating against self peptides. And granted, these are cells that are in a kind of aberrant state, but I don't know, Glee1 is popping off in a lot of cells in my body. So if you want to rile up the anti-vax crowd, you say myocarditis with coronavirus. In the case of vaccination against fibrosis, you might have to worry about a whole other spate of issues. So yes, as you said, needs to be verified, validated. The safety needs to be really, really specced out in a lot of preclinical models before we go with anything like this. But I think what we're talking about today is concept and how we're in a really shifting landscape in terms of how we treat disease, moving really well past this pharmacological paradigm toward a cell-based therapy that's not just, building blocks, regenerative. It's really about using cells as smart therapies. And I'm just happy to be alive in this era, Arun, because I'll live a few years longer, hopefully.
Hey man, that's the dream. And speaking of concept, we're not really talking about a disease related context in this next example. This is a very conceptual story, but in another way. This is super cool, very wild kind of stuff. I actually saw this story as it was unfolding online via Twitter, of course. I'm on Twitter, if you didn't know. Uh Nikki Ebasuya, who actually was one of the former guests on our show. She posted this as a preprint article. The title of this paper is it's a Nature Communications paper. Optogenetic control of apical constriction induces synthetic morphogenesis in mammalian tissues. This is really wild, and it's a synthetic bio paper, and perhaps a nice lead in to our guest on the show today, Dr. Jun Wu. Basically, what the Ebsuya lab here, and the first author here is Guillermo Martinezara. The first, the lab here was manipulating certain signaling pathways and certain growth pathways in cells, in particular, maiden Darby canine kidney or MDCK cells, to really control how they grow, and they specifically controlled the motion and the constriction, the motility of these cells using light optogenetics, as we know, as a really amazing technology that's been pioneered by some folks like Carl Deisseroth at Stanford and others as well. So this is a unique approach to manipulate three-dimensional shapes of mammalian tissues. And the name of this tool, which is a really fun name too, is called OptoShroom 3, an optogenetic tool that can achieve fast spatiotemporal control of apical constriction and mammalian epithelia. And so that's what they did here. They made this custom MDCK cell line that has built-in optogenetic controls, manipulated it using light, and they were able to show that when they shone light on these custom cells, it forced apical constriction, actually enhanced the folding of the epithelial sheet colonies on soft gels, and then they applied it. This is the really cool thing. They applied this optogenetic technology of being able to manipulate cell motion to organoids, to both mouse and human neural organoids. And just by shining a light on these organoids, these custom genetically modified organoids, it caused a thickening of the neuroepithelial, apical lumen reduction in optic vesicles, flattening in neuroectodermal tissues. And I can talk about this forever, but this is one of those studies where you just have to look at the videos, okay? The Embassy Lab, Mickey in particular, actually posted a really, really cool video on Twitter. I very much encourage you to check this out. That's actually why I decided to pick this paper because that video just blew my mind of how you can just shine a light and then you can just control the size and shape of an organoid in real time. It's wild to see. So check that out. Take a look, look at that particular video and check out this paper as well. It's a really cool synthetic biology approach and application more in the realm of developmental biology.
Yeah, this jumped out at me. The first line of the abstract: the emerging field of synthetic developmental biology proposes bottom-up approaches to examine the contribution of each cellular process to complex morphogenesis. I mean, that says it. We've flipped the whole thing on its head. Synthetic biology just this year, the innovations, Arun, are really mind blowing because it just changed now the way you even think about approaching your experiments. And obviously, Abasila was way ahead of the game here because this story's been brewing. But yeah, the idea of capitalizing on this kind of building block approach, uh, everything's converging. Optogenics, the cell printing, and the synthetic bio. It's a brave new world, Arun. And again, just happy to be alive in science. So great. We got to talk to Jun Wu about this one.
Absolutely. And I can see the application that this technology might have for his work. I don't know if you can have some sort of genetically customized or a blastoid that has these optogenetic drivers. Maybe you can control blastoid development just by shining a light on it, which is mind blowing to me. Science fiction.
Well, we're there. Get to that in a minute. Before we do, though, a quick message from STEMCELL Technologies. Looking for more information on organoids? Download STEMCELL Technologies' new ebook on organoid research techniques, developed in collaboration with Wiley Publishing. This essential knowledge briefing details the evolution of organoid technologies from discovery to application, including discussion of key milestones and advances of the technology. A review of key publications and annotated reading lists provide further background on many of the topics covered. Download your copy at www.stemcell.com/organoid ebook. All right, everybody. On the show today, we have with us a special guest, Dr. Jun Wu, who is assistant professor and endowed scholar at UT Southwestern Medical Center. Also, a NICEF Robertson investigator. Devastating news from the NICEF about Susan Solomon a few weeks back. Lost a really shining light in the field. Her legacy is strong and will be remembered. And we can talk with Dr. Wu about her impact and the NICEF's impact on his research trajectory in this interview. But before we get to that, the Wu Lab uses interspecies chimeras to study fundamental biology, conserved and divergent developmental programs, determination of body and organ size, species barriers, and cancer resistance. The lab also works to develop new applications for regenerative medicine. Dr. Wu, thank you so much for joining us today.
Thank you for the opportunity.
All right. Well, getting started, interspecies chimerism, it's part of like the human cultural narrative now going back millennia. We're talking about mythical implications there. So thinking about the actual scientific possibility, like in reality of chimerism, has that kind of mythical overtone to it. But at the end of the day, the practicality of chimerism between species depends on a whole number of factors, one of which is developmental compatibility, right? Approaching 2 decades ago now, I made some naive attempts for my doctoral work to pair human embryonic stem cells with mouse embryos, and it failed spectacularly, although I think it was an informative failure. But we've learned a lot since those days, in large part due to your own work. So give us a brief review regarding the state of the art. What kind of interspecies chimeras have been attempted successfully to date? What are the boundaries and limitations to generating chimeras of all sorts?
So interspecies chimeras are research actually has been going on for several decades already. And then before the arrival of proponent stem cells, specifically the embryonic stem cells, researchers in the past often graft tissues different developmental stages from one species into another species. Some of the application can be used to study the developmental compatibility and also some of the features that are specific to a particular species. For example, between quail and duck, researchers have been using the interspecies camera system to study the beak development, including others as well. With the arrival of embryonic stem cells and proponent stem cells, that made the generation of interspecies much easier. So now we don't have to isolate embryonic cells, which is oftentimes very difficult and technically challenging. But now using embryonic stem cell, because these cells can be propagated in vitro very easily, and they can retain the same potential for a long period of time, we can, and on top of that, we can modify them to whatever way we want, introducing mutations, introducing genes that are not belong to that particular species. And then we can use these cells, inject them into early embryos. And during the formation of the chimeras, some of the cells will contribute to different tissues, and some of the cells will contribute to the germline. With the same species, the germline transmission is very important for generation of transgenic mouse models of human diseases for a number of years. With regard to interspecies chimeras using proponent stem cells, there have been several attempts. Initially, with rodent species, for example, between rat and mice, and between wood mice and mice. The initial attempt was successful. We could generate rat mice chimeras and also wood mice and mice chimeras. In some of the chimeras, you can see a robust contribution of the cells from the donor species into the host species, in this case, the mice as a host species. So largely, interspecies chimeras between rodents are more successful than interspecies between more evolutionary distant species, such as if you're using human cells, injecting human proponent stem cells into animal embryos, the contribution was very limited. And I still remember reading your paper, I believe it's in 2006. That was actually one of the inspirations for me to get into this field, is to how can we solve the problem? How can we overcome the early developmental barrier, so that we can improve the human cell contributions in the animal host, with the ultimate goal of generating human tissues and cells and organs in the animal to overcome this worldwide shortages of the donor cells and tissues and organs.
Well, I gotta say, you've probably just made Daylon's month, Daylon's year, just based on the smile that's on his face right now after mentioning that he was part of your inspiration for getting into the field. So that's very exciting. But certainly, pluripotent stem cells have changed the game in more ways than one, whether it's working with the interspecies chimera technology that you're talking about, but also most recently, there are certainly a couple of bombshell studies that have dropped in the early developmental modeling field that have really caught the public's eye, both the general and scientific public too. You're of course aware of these two studies released by the Jacob Hanna and Magdalena Zernicka Goetz labs, describing these so-called synthetic embryos derived not from the fertilization of sperm and egg, but from mouse pluripotent stem cells cultures in these customized conditions, such as these roller cultures that Dr. Hanna's group was pushing forward. What are your fresh thoughts on these brand new ex vivo models, although they've been in the works for a while now, but just published, these new ex vivo models of mammalian development, these synthetic embryos? And what ultimately do you think is their downstream potential?
First of all, I think these two studies are milestone studies in the stem cell field. Although I have to see that I have heard Magda was talking about this result for a couple of times before the publication. So whenever I heard her talking about this, I was out. Just the fact that you can use pure cultured stem cells to generate embryo that exhibit a milestone developmental process. For example, a beating heart and a neural tube. That is, in my opinion, that is remarkable. Although there is still a long way to go, as you can see, the models are not perfect and the efficiency is not not very high, but I think it's a great start for something truly spectacular. There are potential applications of these is now we know that with the roller culture we can push the development of these synthetic embryos a little bit further. We not only can generate specific cell type, but we can generate early embryonic tissues. And one of the potential applications is we can isolate the progenitor from these early progenitor tissues, and maybe those cells can serve as a better starting cells for differentiating into fully functional cell types in vitro. But as you can imagine, with the field developing further, we can push this limit further by developing mouse embryos, maybe in the future human embryo-like structures into a later stage, so that we can use some of the fetal tissues or fetal organs for therapeutic purposes. Yes, I think both studies are truly remarkable.
Truly remarkable. Bombshell studies. And I like to think that, like you, Jacob Hanna and Magdalena Zernica Goetz were inspired by my early work. So we'll have to send them links to all of my papers and see if they can they can make of that. But on a more serious note, a big part of what has made ex vivo developmental studies possible is the establishment of cell culture conditions that are able to maintain establish and maintain those earliest lineages, right? And part of your work has focused on developing conditions that foster cells with this expanded capacity for interspecies chimerism. So it seems like a really good alignment with this synthetic embryo story. Do you think refining the cell input, by developing more refined culture conditions for the input for these synthetic embryos can improve the developmental fidelity and the success rates of like true ordered gastrulation, et cetera? How do you think we're gonna improve it? I know you just alluded to it, but maybe you could put a finer point on it.
Sure, I think starting cell is absolutely important for both of the applications for chimeras as well as for stem cell embryo models. And we for the past few decades we have been studying a lot, focusing on this proponent stem cells or embryonic stem cells, specifically in mice. As you know, there is a very stringent test for proponency. Called tetraploid complementation. So that has been developed in 20 years ago about also, and then we know that embryonic stem cell can generate an entire animal adult organism after injecting them into tetraploid blastocyst, suggesting that these ES cells we culture in vitro has the potential to do that. But what's lacking in the field is that the understanding or a better culture condition for the actual embryonic stem cells. Although Janet Rosan published the trophoblast stem cell, the first trophoblast stem cells from mouse embryos in 1998, and also in the link cells, the actual embryonic stem cells in 2005. We haven't made much progress in improving those two different type of cells, actual embryonic stem cells. So that resulting in the inability in the past to generate more complete, more functional embryo models. So what Magda and also Jacob did in these two papers is that they use a different strategy. They use transcription factors, and by using transcription factors that are specifically expressed in these two actual embryonic tissues, they kind of force the cells, starting from naive proponent stem cells, into become a more functional trophectoderm or more functional parental endoderm that allow them to assemble this synthetic embryo or stem cell models, embryo models, to go further than we previously couldn't go. And I believe that the development culture conditions for a better culture conditions for actual embryonic stem cells will be more important in the next few years to make the embryo models or make the interspecies chimeras more, to overcome some of the current barriers that we will not be able to.
Yeah, I think it's a critically important thing to consider optimal culture conditions because we're talking about something that's so intricately regulated in early development. And I think another model system where, optimal culture is really critical are these, blastoids that your lab actually developed recently, these human blastocyst like structures, blastoids, quote unquote, that are generated from human naive pluripotent stem cells. And this is, of course, highlighted in your incredible Nature paper that came out last year that showed remarkable similarity between these true human blastocysts and blastoids, between true de novo or truly derived human blastocysts and these artificial blastoids. So tell us where these things stand right now with this blastoid technology that you've pioneered. How is your group improving on it? What are some of the optimal culture conditions that you're focused on developing for the next stage of these blastoids? Tell us about what's going on with the blastoids currently in your lab.
Sure. I think before I talk about the blastoid, I need to emphasize that the blastoid model or any other stem cell embryo models are not equivalent to the embryos. Although many of this, by reading the paper, sometimes you believe these models are exactly the copy of the human embryo or a mouse embryo. They're not. There are still a lot of differences between them, and I think we should spend some effort in understanding the differences rather than try to advertise that these models basically the exactly the same as a human as embryos. So we first published the blastoid model last year. As you probably know, they are not perfect, so the efficiency was not very good. We ended up with around 15% of efficiency across different cell lines, and by single cell RNC, we identify there is off-targeting cell types found in these blastoid models. But we also, through other assays, we also show that these blastoid models are very similar to human blastocysts in terms of the size, the composition of the lineages, and also this total cell number and cell number in each different lineages. They are very similar. And you may also know there are several other papers published after us, and then obviously with improved efficiency, improved methods, and now especially with the study with the Nicholas Riveron's group, they were able to show that you can generate a human blastoid starting with naive human pluripotent stem cell at an efficiency around 70% to 80%. I think that's very very great. And they also show that some of the blastoid in general, at the transcriptional level, they mimic the human blastocyst. And we have also improved in my lab. We have recently improved the efficiency and also the fidelity of our own models using our own strategy. We use a two-step strategy, and Nicholas Riveron and also Deguo, they use one-step strategy. But at the end of the differentiation, we found that our newly improved blastoid model also around 80 to 90% of the efficiency, and the fidelity is greatly improved. What we're focusing on is to use this blastoid to understand implantation and also the crosstalk between blastoid and the endometrial cells, such as endometrial epithelial cells and endometrial stroma cells. Specifically, we were focusing on the stroma cells because once the blastoid or blastocyst, if they ever implant into the uterus, the cell type that they interact the most is the stroma cells. And so we established a co-culture system by culturing the blastoid together with the endometrial stroma cells that we isolated from the patient. And by doing that, we were able to find that the stroma cells can promote the differentiation of the trophoblast and making them more easier to become syncytial trophoblast containing multi-nucleus than the conditions that without these stroma cells. That's just one of the examples, but there are other examples that we can use blastoid for various different applications. But during this study, we found that blastoid and blastocysts are very different in terms of their attachment rate, in terms of their growth on top of the stroma cells. We found that the human blastocysts, they grow more robustly than our blastoid, and the cells proliferate much faster the blastoid. So something that we still try to figure out the reason why. Obviously, we cannot do these type of experiments here in my own lab. We collaborated with a group in Colorado. It's called a CCRM. So they have a facility they can use human blastocysts to do research in this type of research.
Yeah, I love that example that you use there, because as someone who's really, got a personal and professional interest in reproductive biology, that's the thing I think that has been overlooked. That end game of the attachment to the endometrial cells was used as kind of a proof. Oh, these are blasts with trophectoderm, and they can model this implantation type phenotype. But what I think, the developmental biologists amongst us and reproductive biologists recognize there is that kicks open the door to this whole black box that was implantation biology. And that there, as you are illustrating, the really important lessons that we can learn, not just about like what the mechanisms there are, but like how these blastoids differ from human blastocysts, and how we can maybe improve implantation for the purpose of better outcomes in the context of assisted reproduction. So it's such a great example of how the real clinical output of this is not just, chimerism or in vitro synthetic embryos, which maybe is the flashy headline. It's real clinical upshot there in a field that really has not had any models to explore this. And I say all that, but getting back to the chimerism, as I just said, we can appreciate that the primary focus right now is application toward, or chimerism at least, and other science, to deciphering the many mysteries of developmental biology. But of course, we've all thought about the idea of utilizing interspecies chimerism as a means of generating human organs, both for clinical study or even transplant, right? That faces many obstacles toward being feasible and practical, of course, but the progress, I think, has been remarkably swift and is still ongoing. I have a vested interest myself, again, in the clinical application of human eggs that have been cultivated in mouse xenografts from cryopreserved ovarian tissue for whatever reasons. You can look at my work for that. But so I'm hopeful, I'm a fan. But you are a scientist who thought very deeply about this. So I'm counting on you to give it to me straight here. Is clinical application of xeno-derived cell products feasible in the current regulatory landscape? Like, can we get it through the FDA?
I I think the challenge there currently at the science and so technology is we're not there yet. Uh, the prospect of generating human tissues in animals are certainly very attractive, uh, especially generate organs that are can be later used for transplant. But talking about science today regarding using interspecies chimera, using xeno-derived tissues and cells, we're not there yet. The reason being is that there's a lot of barrier uh, during early development that prevent the efficient contribution of the human cells in the animal host that we are currently using. For example, pigs. Even though pig and human are very similar in terms of their organ size, physiology, but they're still very different in the evolutionary tree. They separated by more than 90 million years in evolution. And we know that evolutionary distance are in a way is a measurement of how difficult we can generate interspecies chimeras. When we use more closely related species, such as mice and rat, we are able to generate more robust chimerism. But with a more evolution distant species, this become extremely difficult. So we're still at this stage that we try to overcome the human cells survival in the early stage of the embryo development in mice and in pigs. So I would say that we're not there yet. So I think I don't know when that will happen, when we can realize the dream, but at least it's not there today. Yeah.
Yeah, with that being said, there have been some very high profile victories recently when using xeno-derived organs. For example, the humanized pig that was genetically optimized to modify its immune system so it could have organs like a heart, for example, that could be transplanted for some duration of time into that patient in Maryland. I mean, that was a big milestone. It's not obviously we're not talking about chimeras in that situation, but we're talking about xeno derived tissues that are being transplanted into people. So certainly there is progress, but it's like what you're alluding to. It's extremely difficult scientifically to do these things for a lot of different reasons. But talking about the long-term goal, it's no secret that one of the long-term goals that we're alluding to, whether we're talking about chimeras and blastoids, is like we're talking about generating human organs to combat the current shortage of organs available for transplantation, and also perhaps to alleviate the black market for organ transplantation. I mean, Jacob Hanna, he directly mentioned this because he's got a startup that's going to be focused on something similar to this area of generating human organs for transplantation purposes. It's a huge need. Now, countering the developmental biology approach is the bioengineering approach, right? I think there's two independent means of thinking about this. You're, of course, a developmental biologist first and foremost, but I think a lot of bioengineers are thinking about perhaps using 3D printing and other these artificial approaches to actually also generate tissues and maybe one day organs for transplant. And certainly, as a developmental biologist, you have your own biases towards taking those chimeric or developmental based approaches. But what are your thoughts on these bioengineering based approaches for generating tissues and organs for transplantation? Or do you think the best bioengineer is the body itself? That's kind of a crude statement, but what do you think?
So since you mentioned the xenotransplantation, I want to comment on that. The xenotransplantation uh, are more promising than the chimera approach currently because that involving the genetically modifying a pig to reduce the immune barrier so that they can be transplanted into humans. There, as you mentioned, there are several preclinical studies on that, and they seems very promising. And that xenotransplantation field has been going on for more than 2 decades, more than 2 decades. And then with the arrival of the CRISPR, now we can more easily make genetically modified pigs than before. I think that really revolutionized this field. Uh, but back to the engineering approach, I still think it's one of the alternative ways that can be potentially possible in the future. The reason being is that if you read carefully about the Jacob Hanna and Magda's paper, I don't know about you guys, but when I was reading it, all of a sudden I realized we actually don't need the cells to go through the full developmental process in order to generate organs and tissues. So they're using naive mouse embryonic stem cell, which is equivalent to about E4.5 mouse aggregate cells. And then they assemble these naive cells overexpressed with CTX2, GATA4 to give rise to these two actual embryonic tissues. They assemble together into an embryo-like structure that the equivalent or similar to E5.5 embryos. And from that point, they culture in vitro and to about E8.5 that give rise to a neural tube and beating heart, suggesting that if we have the right cell type, maybe we can assemble at a different developmental stage of different tissues, different organs. So again, back to my original thinking is that the starting cell is most critical. If we can, for example, isolate or culture a progenitor cells for the heart, let's say, and also the surrounding cells that are supporting cells, if we can culture them, we show that they are more faithfully recapitulating what we observe in vivo. Maybe using engineer approach, we can put them together and assemble a heart de novo in vitro without going through the entire developmental process. I think that's what I learned from these two papers. What I kind of maybe these two papers suggest that we really don't need starting from the zygote, fertilized egg. We can assemble tissues and embryos at different stage of the development if we have this right cell type.
Yeah, that we've talked about it before or mentioned it at least on the show. It's such an exciting time to be in science, but specifically as a developmental biologist with stem cells, because there's this element of convergence, right? From all ends, bioengineering, developmental biology, genetics, CRISPR. it seems like everybody is on the same track. And as you said, we're kind of bypassing the need even to go from an embryonic rudiment. You could just get the organ enlage and yeah, necessitating the bioprinting, which is finally maturing. It's so awesome, this kind of scientific singularity we're in the midst of, or at least it seems. But as you're kind of getting to there, that would avoid a whole kind of bioethical maelstrom, right? I mean, at the front end of the chat, we were talking about what the scope, what kind of chimeras have been made, and you had another huge paper, one amongst the many feathers in your cap, looking at human macaque chimeric embryos in like a kind of peri-implantation phase ex vivo. That was in Cell about a year ago. So clearly the experimental capacity to intermingle early cell lineages between human and non-human primates, it's there. It's feasible. And we've talked on the show before about ISSCR guidelines on interspecies chimerism and ex vivo embryo development. So there is kind of guideposts there on how we all agree on what the limits should be. So we don't need to talk about that, but your insight, I think, is unique. I mean, you've had arguably the most firsthand experience with navigating a really tough bioethical landscape with the blastoids, with the interspecies. It seems like everything you touch, you got to have a bioethicist weigh in on it. So can you give us an idea of what the boundaries here are? Maybe even it would be informative for you to share what experiments are out of the question, for example, and what it's like to have to really navigate all these committees at UT. Like, what's it like to be in such a hotbed of controversy?
Yeah, we'll be happy to talk about that. So for the human monkey ex vivo chimera work, it's not done here at UT. So it's done in my previous institute when I was working with Juan Carlos Isbisua Belmonte at the Salk Institute. We were collaborating with the lab in China. They have one of the largest macaque colonies in the world. So we saw that we could team up with them to examine the contribution of human cells into non-human primate species in the early embryo. The reason we were doing that experiment is not we want to use macaque as a host species for generating human organs. The only reason was to examine whether human cells has a better chance of contributing to the early embryos of a species that are close related to human, to answer the question whether evolutionary distance plays a role into interspecies chimerism, which we have done that with the rodent, but we don't have much of the information with the human. And by doing that, we realized that with a limited experimental setting that we were in, the human cell indeed can contribute at a higher level, at least a seemingly higher level in a early macaque embryos, cultured ex vivo, and also by single cell RNA-seq analysis, we were able to identify these similarities and differences of putting the human cell inside macaque embryos and compare them with mouse embryos in the future. Maybe can help us to understand the differences between species. So here at UT Southwestern, my lab is mainly focused on using mice as a host to study the interspecies barriers. Actually, most of the project in my lab is to use in vitro co-culture system between species at different stages of development. Since the start of my lab, we have established a co-culture system between pluripotent stem cells from different species. I'm hopeful that by using this in vitro model, instead of using in vivo embryos, we can help us to understand the species barriers as xenogenic barriers, and it also can develop strategy to overcome these barriers. For example, by co-culturing human and mouse pluripotent stem cells, we found that the mouse cells are become more aggressive. They're starting to attack human cells in the process called cell-cell competition. In addition, but we also identify there are several other barriers. For example, incompatibility between cell-cell adhesion between species. We're actively working in that direction to overcome this incompatibility of cell-cell adhesion. we almost there, but we're still trying to figure out, try to perform the chimera experiment to prove this our strategy can actually work. We have successfully overcome this competition in vitro, but the next step will be to inject human cells after modifying the cell-cell adhesion in compatibility into the early mouse embryos to see whether they can improve chimerism. Yeah, so because of using the in vitro culture, we're not have the similar kind of ethical barriers that would if we were using the early embryos for this work. With the blastoid, I think there's a different ethical challenges, because when we first generate this, or we first wanted to generate this blastocyst-like structures, we talked to the school committee at the UT Southwestern. They don't know how to regulate this. They don't know what, because at that time there is no guideline to regulate this type of research. And luckily at the time there's a stem cell report paper headed by Nicholas Rimeron and also Martin Pera and several other leaders in the field. They came up with a recommendation before the ICCR released their official guidelines last year that help us, help guide this line of research. So I sent that article to our school committee. They read it and they agree. It's better to regulate with more precaution. So at the time they treat it as a human embryo, but obviously they are not human embryo, and I show them that there is a differences. So they allow me to do this type of work in UT Southwestern. But as you know, in Texas, we're not allowed to work directly with human blastocysts. But I'm very grateful and also very happy that they allowed me to continue this blastoid work and even to this day. So yeah.
Yeah, there's a lot of considerations. You certainly have to consider where you are, in terms of being in Texas. And I think one of the coolest things about the work that you do is how collaborative it is. Like you mentioned, you're collaborating with folks in Colorado, across the world. And that's exciting in part because you have, different regulatory considerations to think about, but also the scientific influences and the scientific collaborations, I think ultimately leads to better work. You mentioned, of course, you are at UT Southwestern, which is a powerhouse medical institution and biomedical research institution. We actually just had a junior colleague from UT Southwestern, Ed Groh, on our show. And we always like to discuss why certain PIs pick the institutions that they pick for starting their new labs. Ed, mentioned that there's a history of UT Southwestern for cutting edge biomedical research. In my own field, there's of course Eric Olson, who has been at UT Southwestern for a very long time and one of the grandfathers of modern cardiovascular biology. And you've had your lab at UT Southwestern for a little while there. So give us your perspective, about what makes UT Southwestern tick as an institution, why it's such a powerhouse, and what do you like about the place?
Absolutely. So there are a couple of reasons that I choose UT Southwestern when I started applying for faculty jobs. One of the reasons is Eric Olson, because he's the chair of my department, and I know him very well, and he's very supportive in stem cell research. And another reason that I moved here is that UT Southwestern is known for studying the mechanism to understand how things work. So I think that's been lacking in a stem cell field, especially the interspecies chimera field. So there's a lot of hope and promises that are being made, but we realize there's many barriers to the success of generating human organs and tissues in animals. But at the time I thought maybe I should choose an institute that surrounded me with people that can help me to understand why it didn't work, and which I think I made the right choice with the colleagues here on campus. They're biochemists, the cell biologists. By talking to them, that expand my thinking and also help me to build this in vitro system to really dig deep into the mechanism, how the interspecies barrier are regulated. By only doing that, we can come up with effective strategies to overcome these barriers. And without that, I don't think that we will realize our dream of generating human organs using this interspecies chimera approach. Although we're still not there yet, but we're making progress one step at a time, overcome the barriers one step at a time. I'm still hopeful that in the future, we can understand the barrier enough so that we can develop effective strategies to really have our ultimate goal realized in the end.
Yeah, for me, and just listening to your answer there, it kind of presented in microcosm of chimerism as a field, kind of the arc of science. In that, yeah, because you said it. UT, what's great about UT Southwestern is a place where they're trying to figure out how things work. And I remember when we were doing the chimerism initially, we were seeing if it worked. But what we did is we did one of the first steps in seeing how something didn't work, right? And I think that's always a first step in science, is figure out how something doesn't work, and then perhaps why it doesn't work. And you've been just pounding away at this for so long that you're getting us to this really unique place in science where you're in a position now to figure out how it can work, how it might work, and ultimately, I'm guessing how it will work. So I'm just excited to have been able to have this conversation, and frankly, I'm still beaming a bit just to consider that you read my paper and it had any input in your thinking. So thanks for that. But before we let you go, a couple of peripheral questions we have for you. First, if you could answer any single scientific question, regardless of your expertise or chosen field, what would that be?
I'm a little bit biased. I want to understand propotency across all mammalian species. That's my ultimate dream, is to derive and generate propotent stem cells from all mammals on Earth. There are over 5,000, I believe, mammals. So as you can imagine, they will take a few more years and a few more trainees together. But that's ultimately my dream, is if we can understand how propotency is regulated across different mammalian species, that will help us getting closer to understand how life is started as a mammal. That's the ultimate question I want to answer.
Wow, modest goals from Dr. Wu. I mean, you thought pluripotency was a thing, but here after this conversation, you're recognizing that it is everything. And lucky for us, we got Jun here, who's going to go after it. Finally, if you were not a scientist, what would you be?
I think I will be writing science fictions. I really love science fictions when I was growing up, so I think that's one of the passion. So if some of the ideas I cannot do it in my own lab, if I'm not a scientist, I probably put in the book, and maybe in the future, some kids or people that are inspired by some of the ideas and maybe can make it work. So.
Well, from a fellow sci-fi geek, I can say I probably speak for Arun here too. We would read that book, my friend. You send us a draft. I would love to walk around in your imagination. And thanks for sharing all these inspiring words. And we hope to have you back after you've broken off a few more species of pluripotency and solved that mystery. Thanks again, Jun. We'll see you soon.
Thank you for the opportunity. Really enjoy the conversation with you guys.
All right, that brings us to the end of this episode. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on Twitter at STEMCELL or via email at info@stemcellpodcast.com with feedback or to suggest guests. Always nice having Dr. Wu on the show. Gets us into that whole other zone, thinking about chimeras, the future of cell-based science. We'll be back with another great guest in a couple weeks. Until then, thank you so much for listening.